Chairperson. Good morning, everyone. It's my honor to stand here, give my speech. Introduction of wood identification to CITES list and China protection species. Advantage and limitation. There are 4 parts. The first one is forward due to the illegal logging and excessive logging and tradition in the illegal timber. So the forest deforestation. I'll give some examples. This picture shows the illegal cutting. Is a Derbogia Cochranitis. This species belong to the CITES list. The picture taken in the Cambodia. And the next picture is illegal cutting, also from Derbogia in the Jianfengling, Hainan Islands in our country. And also illegal cutting. Also, same species, the big trees have been stolen in the Guangdong province in our country. As you know, the international region trade law and convention have been launched. For example, its famous CITES and Lazy Action Amendment. European Union Timber Regulation and Chinese Regulation, etc. As you know, the CITES is exciting to the timber species. For example, in the 2010, the numbers of the timber species controlled by CITES is about 111. But 2013 is increasing too fast. The numbers is 2,048. New progress of CITES list the timber also added 3 species provided by 2 countries. So, identification of wood species and origin is very important things. The part 2 is introduce wood ID methods. Usually we use the 4 methods. First one is use the microstructure. Everyone know that is we can use the handland. Then observe the transverse surface. You can know which one is conifer and which one is hardwood. And we also can observe the parenchyma, so you can do some ID. We also use the tangential surface. We can find some storage range is have and no. Then we compare with the wood collection, such as color, odor, hardwood and softwood, grain strain, grain, vessel, parenchyma, grain, density and hardness, and so on. For example, the taxi species is listed by CITES, but another one, procedural taxi, is not. So we can use the microstructure identify them. The 2 methods is micro and macro structure. We usually use this method. So you can see the 15 and 20 macro section can be cut, and we use the 3 direction, transversion, tangential, and radial section. Then staining, dehanging made the microscope slice. The tissues arrangement and cell characteristics are vessel parenchyma ring observed and the microscope by observed. Then compare with the wood collection and their section. Actually, to the genus and subgenus, this method. And another method is physical and chemistry. You can see this picture show the blue colors. This is from Protocarpus centennials. They have fluorescence. And the third one is use this picture show the Acularia and centennials. These are famous wood species. So we can use the combustion method, then get very mild smell, then adding them. The third one is use the software. The database of Chinese conifer and hardwood have been established, and also established the database Latin American tropical timber and Africa tropical wood and Southeast tropical timber. But we use the wood anatomical method have limitations because without leaves, flowers, seeds, so we can't with no species and no origin. So the new method for the wood ID should be developing. The new method for the wood ID. Now in our academy, we use the DNA barcodings, the method. Then the experimental is argan wood. Just told you the Acularia synthesis is in our Chinese species. It's belong to the is controlled by CITES and our national protection the flower class 2. So we use the fresh and the drying and the different temperature the wood disc, and also use the fresh leaves. So we make wood powder for the DNA extraction. So the results indicate use the nuclear DNA, so we can distinguish the Acularia synthesis from another species in the same genus. And also another experimental, we use the aged and archaeological Papyrus Europhaetica wood. We use the different story times, used more than 30, 55, 8, and more than 3,000 years. So the results indicate we can use the DNA method and distinguish this species and another Papyrus genus, the species. I also do another research. This species also from Derbogia, also controlled by CITES conversion. We use the wood samples and the leaves. So we use the 2 DNA methods. So the results indicated, and use the DNA method we can ID these 3 species. So this used DNA method is just the beginning. So I believe the global efforts are needed to improve the method for the DNA extracting from dried and aged wood. And the second one, the works for the win-win cooperation on the sampling and the database development. Fourth one, future works. Future works have 5 points. Point 1 is the wood collection exchange. This picture shows our Academy wood collection. This have a long history. Established in 1928, biggest one in China. They have 500 species. So we want to, in the future, active exchange of wood samples with different institute and university. And another wood ID method should be developing. For example, stable isotable. So in our country, no solid progress, but in Germany and another country have been done. Another method, new method, is NIR spectral. So we can find the timber species and origin. Several small model and database established in our academy. Also, we do a lot of training course on wood ID. For example, more than 10 times, such as Xiamen customers and Hong Kong and Macau CITES office and so on. This picture shows China, Germany joint training course on the CITES timber identification in the last year in our academy. So the training course, the purpose is improve capacity of quick prognosis. And point 4, domestic collaboration. Yeah, the first one is we got national project supported by State Forest Administration. And the title is New Technology of Wood ID for the endangered tropical species. And we are having done the domestic laboratory collaboration. Our academy with the Chinese Academy of Science and Guangxi University and Shanghai and Anhui and another unit and university. Point 5 is international cooperation. We are joined the international project supported by BI, Biodiversity International. The title is Identification of Wood Species and Origin. The picture shows the project steering committee, the meeting. And this picture shows the Asia Pacific region workshop. It's held in the Beijing in the 2013 years. The title also is Identification of the timber species and regions. This picture shows our research group. Yeah, we are like wood very much. Wood is good. That's all.